Hello dear students. In last lecture we studied about the central dogma for flow of information. In that we studied that the information flow is from DNA to RNA and RNA to proteins. And this information, that is DNA to RNA, is called as transcription. So in last lecture we studied about the definition of transcription, and then we studied the structure of a transcription unit. So you must have understood the structure of this transcription unit. If you see this transcription unit, you can see that the complete DNA doesn't undergo transcription. Only a specific part which is having the promoter and terminator along with the structural gene, that is histone, which is called as transcription unit, that undergoes the transcription. And on a DNA there may be many transcription units. So this transcription unit, last time I told you, DNA is having two strands and both the strands are complementary to each other. And we studied that both the strands do not undergo transcription. Only one strand undergoes transcription, which is called as the template strand. And the strand which is not undergoing transcription, but it is just similar to the transcript produced, that is called coding strand. All the references on the transcription unit are made with reference to coding strand. That's why the coding strand is also called as sense strand, and the template strand is called as antisense strand. Okay, keep it in mind. So in today's lecture we will be studying the complete process of transcription, and then we'll study how the transcription unit in a prokaryotic cell is different from a eukaryotic cell, and how the process of the transcription, though it is almost similar in both prokaryotes and eukaryotes, but there is some complexity in eukaryotes. We will study that. So let's study the process of transcription. Let us now study the process of transcription. The enzyme responsible for the transcription process is DNA-dependent RNA polymerase. So this DNA-dependent RNA polymerase, it has got some specialities. First is it will always polymerize from 5' to 3' polarity. Number one. Second, it does not require any primer. Third, it will unwind and do the elongation process both. So, when this RNA polymerase finds the promoter, there is an initiation factor which is called as the sigma factor. It gets binded to the RNA polymerase at the promoter site, and the process of initiation begins. So the first process in transcription is called as initiation. So in the initiation process, this RNA polymerase along with the initiation factor, that is sigma factor, binds to the promoter site and it begins the process. What it will do? It will first unwind the double-stranded DNA, around 25 to 30 base pairs. Okay, and then based on the template strand, that is the strand showing the polarity 3' to 5', it will start making a transcript based on the template strand. That strand which is showing the polarity 3' to 5' will become the template strand or the antisense strand. Okay. Now, this RNA polymerase DNA dependent RNA polymerase, it will show the elongation process just like the DNA replication. It will get the charged polynucleotides, and depending upon the basis on the template strand, it will keep on adding the new new nucleotides. Okay, and you know ATCG. A, the complementary is T, but in the RNA, instead of T, it will be uracil, U, and C, it will be G. A will bind with uracil, and T will bind with adenine. C will bind with the decytosine, will bind with guanine, and G, that is guanine, will bind with C, cytosine. So based on that, there will be the process, second step, which is called as elongation. Okay. Now, a point will come, that is the termination point. So at the termination point, there will be this sigma factor will get removed from the DNA polymerase, as RNA polymerase, and a termination factor, that is termination factor which is called as rho, that will get binded to it, and this RNA which is transcribed is released off as a new transcript. So these are the three steps, that is initiation, elongation and termination. Okay, so these three steps are similar in both prokaryotic as well as eukaryotic, but there are some differences. Let's now find out the differences between prokaryotic transcription and eukaryotic transcription. The process of transcription which I explained is actually, this one is shown by the prokaryotes. Before studying the eukaryotic transcription, I must tell you there is a difference in the transcription unit of prokaryotes and eukaryotes. The main difference is, in prokaryotes, since there is no nucleus, all the process takes place in the cytosol, that is in the cytoplasm. Okay, but in eukaryotes, there is a nucleus. The process of transcription takes place in the nucleus and then it is released into the cytoplasm. So in prokaryotes, transcription and translation, they go one after the other and simultaneously. But in case of eukaryotes, it's first the transcription followed by the transmission to the cytoplasm, where the translation will begin. Okay. Second, in case of prokaryotes, the transcription unit is polycistronic. Polycistronic means there are many cistrons in between the promoter and the terminator. So promoter, cistron 1, cistron 2, cistron 3 and so on, and ultimately terminator. So many mRNA transcripts are produced, different types, in one complete transcription. And these transcripts, that is RNA, which are produced, they are ready for the translation process. Okay. So in prokaryotes, the transcription unit is polycistronic. Polycistronic means many cistrons on one transcription unit. Also, all the, you can say the sites which are produced, they are meaningful information. That is, whatever RNA is produced, that is a final transcript and that is ready for the process of translation. But in case of eukaryotes, the process is quite complex. First process, The transcription unit in eukaryotes is monocistronic, which means one transcription unit will have promoter, cistron and terminator. Then next we'll again have promoter, cistron 2 and then terminator. Okay. So one transcription unit will code for only one structural gene. Okay. Not only that, that structural gene which is present, it will produce an mRNA, which will produce an RNA, which will have many units which are not meaningful. Or you can say the transcript which is produced is heterogeneous nuclear RNA. It will have alternate introns and exons, which I'll explain. So, there is lot of processing required in case of the transcription in eukaryotes. So, let us study the complexity in eukaryotic transcription. Basically, the processing required after these three steps. Let us now study the process of transcription in eukaryotes. The process is almost the same in the beginning. That is, it also includes initiation, elongation and termination. After termination, the transcript which is produced is called hnRNA, heterogeneous nuclear RNA, which needs to be processed. I will come to the processing later on. There is one more complexity. In case of prokaryotes, there is only one type of RNA polymerase, which does the whole process. But in case of eukaryotic cells, there are three types of RNA polymerases. They are RNA polymerase 1, which will transcribe only rRNA, that is ribosomal RNA. Whereas RNA polymerase 2 is the main enzyme which is responsible for transcribing the mRNA, which is initially hnRNA, that is heterogeneous nuclear RNA. And there is RNA polymerase 3, which transcribes for tRNA, that is transfer RNA, 5srRNA, that is small ribosomal nuclear RNA, and snRNA, small nuclear RNA. Okay, so this is one complexity. Now, we are mainly concerned with mRNA. mRNA is initially transcribed in the form of a transcript which is called as hnRNA, heterogeneous nuclear RNA. In heterogeneous nuclear RNA, it will not undergo translation unless it undergoes the process of processing. And processing includes three steps, which are capping. Capping means addition of guanine triphosphate at the 5' end of the hnRNA. Okay, so that is capping. After capping, there is splicing. In this hnRNA, there are different parts. You can see black and red part. These red parts are called introns, which are not meaningful information. They are transcribed, but it is not the information which is required by the cell. And red part, that is intron, and black parts which are exon, which are meaningful information. So, before you can say the mRNA is released, these introns have to be removed off by the process called as splicing. There are certain factors called as spliceosomes which will cut these introns, and a transcript will be produced which will be having all exons. These exons will be binded together and introns will be removed. This is called splicing. And third part, that is polyadenylation. That is around 200 adenyl units are added at the 3' end. A tail is added. It is also called as tailing. Many adenine units are added to the 3' end of this transcript. So these three processes, that is capping, splicing and polyadenylation, after these are completed, then whatever transcript is produced, that is known as mRNA, messenger RNA. And this messenger RNA only will be released from the nucleus into the cytoplasm. Okay, so we can see that there is complexity in eukaryotes. So please go through this transcription in prokaryotes and transcription in eukaryotes. It's a very important process and it is very important from exam point of view also. So practice the diagrams both for prokaryotes as well as eukaryotes. Okay, thank you.